Hi everyone, I am Sher Li from NTU and I'll be presenting my poster, which is titled Ermin deficiency as a model for understanding early mechanisms of abnormal myelination in neurodegeneration. So my project focuses on myelination or abnormal myelination, which has been hypothesized to be a key driver of several neurodegenerative conditions, including Alzheimer's disease, Huntington's disease, as well as multiple sclerosis. So I'll be studying the Ermin gene, which is expressed specifically in mature oligodendrocytes during late central nervous system myelination. And this forms the myelin around axons of the brain and spinal cord. So when there is Ermin deficiency, it has been observed that the myelin undergoes fragmentation and degeneration. And so it is hypothesized to play a role in actin filament cross-linking, which promotes cytoskeleton remodeling and oligodendrocyte arborization. So for my project, I have Ermin deficient mice, which is a model developed by Prof. Mahmoud Puladi, who's currently at UBC. So this is a CRISPR-Cas9 knockout mouse with a deletion of 201 base pairs at the promoter and 5' untranslated region of the Ermin gene. And that causes a truncation of the gene and therefore is not expressed. So for my project, I have Ermin knockout mice, homozygous knockouts, as well as heterozygous mice, as well as wild-type mice, and male and female mice from both 1 month old and 3 month old age groups. So I conducted analysis on bulk RNA sequencing data from the corpus callosum of these samples. Yep, and the corpus callosum contains a lot of axons because it is the largest white matter fiber bundle in the brain. So for my analysis, first I conducted a differential gene expression analysis to look across genotype if there are differences caused by Ermin deficiency. And then I also conducted a weighted gene co-expression network analysis, which looks at networks of genes that have similar patterns of expression, suggesting that they have similar regulatory processes. And then I also did a differential co-expression analysis, which looks at how these co-expressed networks differ across the genotype condition. So from differential gene expression analysis, the key takeaway would be that at 3 months, 3 months old in both male and female samples, there are like widespread changes, including those seen in protein processing. But more importantly, there are changes seen in synaptic signaling, particularly in the female mice, as well as cytoskeleton and microtubule organization seen in the male mice, which provides some information on some of the pathways that are affected in Ermin deficient mice. And looking at the co-expressed networks of genes in both 1 month old and 3 month old groups, we see that there are there's regulation of the genes that are associated with neuron enrichment and myelin assembly, which relates back to Ermin function, but also related to microtubule bundle formation. And in 3 months, we see that there are networks of genes related to synaptic signaling and cell projection, the extracellular matrix. And just as a sanity check, again, the myelination and neuron enrichment is also seen to be regulated at 3 months. So looking at differential co-expression to compare how the gene regulation could differ across genotype conditions, from 1 month old, we already see that the mice have differential co-expression in synaptic signaling and neuron differentiation, suggesting that these processes are being affected from a very early age. And in 3-month-old mice, we also see that the differential co-expression occurs with genes associated with gliogenesis and neuron enrichment. So for my project so far, the findings would be that the early molecular mechanisms of abnormal myelination are variable with dependence on age and sex, as there are differences in male and female mice. And some of the terms that some of the genes that are being affected are associated with cytoskeletal organization and synaptic signaling, suggesting that these processes are affected in Ermin deficiency and could affect both oligodendrocytes and neurons. And finally, we observed some differences between male and female mice, which could relate back to clinical phenotypes as the diseases associated with myelin deficiencies or myelin defects such as multiple sclerosis also tend to have sex-dependent differences. So for future work, I will be looking at the cell type specific effects of Ermin deficiency, specifically on oligodendrocytes where they are expressed. And I'll be looking further into the roles of cytoskeletal organization and synaptic signaling and how these are regulated in Ermin deficiency across age and sex. And I will be also doing a comparison between Ermin deficiency and other myelin-associated genes or genes associated with disease. Yep. So I would like to thank my lab. I'm from Prof. Sarah Langley's lab, and all my lab members, as well as collaborators, especially Prof. Mahmood, who has provided us with the mice. And was also a very big part of this project. And thank you to the conference organizers as well for the opportunity. Thank you for listening.